Good morning, students. Welcome to Medicinal Chemistry classroom. Today's topic is antibiotics, which is a very important topic. So first, we will see the definition of an antibiotic. Antibiotic is a chemical compound that inhibits or abolishes the growth of microorganisms such as bacteria, fungi, or protozoa. Actually, they are chemical compounds derived from or produced by microorganisms such as fungi, actinomycetes, bacteria, yeast, or molds. And this definition has now been widened to include synthetic antibacterial chemotherapeutic agents such as sulfonamides and quinolones. It also includes any agent with biological activity against living organisms. Moving on to the classification, antibiotics are classified into four types. First one, based on chemical structure. Second one, based on pharmacological activity. Third one, based on spectrum of activity. And fourth one, based on staining methods. First, we will see classification based on chemical structure. First one is beta-lactam antibiotics. Examples: penicillin, cephalosporins. Second one, aminoglycoside antibiotics. Examples: streptomycin, neomycin, paromomycin, kanamycin, amikacin, gentamicin, tobramycin, netilmycin, zizomycin, spectinomycin. Third one, tetracyclines. Examples: tetracycline, chlortetracycline, rolytetracycline, oxytetracycline, metacycline, dimeclocycline, meclocycline, doxycycline, minocycline. Fourth one, macrolide antibiotics. Examples: erythromycin, clarithromycin, azithromycin, dyrithromycin. Next one is lincomycins. Example: lincomycin and clindamycin. Polypeptide antibiotics. Examples: vancomycin, teicoplanin, bacitracin, polymyxin B, colistin, gramicidin. Last one, miscellaneous. Example: chloramphenicol, novobiosine, mupirocin, quinaprisin, fusidic acid. So, the first classification is based on the chemical structure of the compound. Next one is classification based on pharmacological activity. First one is antifungal antibiotics. In that, pollines, example: amphotericin B, nystatin, natamycin. Others, example: griseofulvin. Second type, anti-cancer antibiotics. Examples: dactinomycin, daunorubicin, doxorubicin, bleomycin, idarubicin, mitomycin, pligamycin, streptozocin, valrubicin. Third one, anti-typhoid antibiotic. Example: chloramphenicol. Fourth one, antidiarrheal antibiotic. Example: colistin. Fifth one, anti-tubercular antibiotics. Examples: rifampicin, rifampicin, cyclosarin, capreomycin. Under classifications, we have two more classifications. The first one is classification based on spectrum of activity, that is broad spectrum antibiotics and narrow spectrum antibiotics. And fourth type of classification is classification based on staining methods. And under that we have antibiotics which are active against gram positive bacteria and those which are active against gram negative bacteria. Okay, so I have given only the first two classifications. First one is classification based on chemical structure of antibiotics, and the second one is classification based on pharmacological activity. Now, move on to the first type of classification, that is beta-lactam antibiotics. So, under beta-lactam antibiotics, we have penicillins and cephalosporins. So, what are beta-lactam antibiotics? They are antibiotics that contain beta-lactam nucleus in its molecular structure, and they are currently employed for chemotherapy and bacterial infections. They include penicillin derivatives, cephalosporins, monobactams, carbapenems, and beta-lactamase inhibitors. So what is a beta-lactam nuclease? As you can see in the structure, it is a cyclic amide. It's a four-membered cyclic amide, and those antibiotics that contain the beta-lactam ring structure, and they constitute the dominant class of antibiotic agents or antibiotics which are currently employed for chemotherapy of bacterial infections. Okay, so penicillins and cephalosporins are the important ones, and we also have monobactams like astrionum. Why they are termed as monobactams? Because and the beta lactam ring is not fused to any other ring, so beta lactam ring is alone in the nucleus, hence the term monobactam. And the second one is carbapenems. So carbapenems examples are imipenem and meropenem. And beta-lactam inhibitors are clavulanic acid, sulbactam, tazobactam, relebactam, etc. Moving on to the mechanism of action of beta-lactam antibiotics. So beta-lactam antibiotics, they are bactericidal and act by inhibiting the synthesis of peptidoglycan layer of bacterial cell wall. So the cell wall of bacteria is essential for the normal growth and development. So peptidoglycan is a heteropolymeric component of the cell wall that provides rigid mechanical stability. The last step in peptidoglycan synthesis, that is transpeptidation, is inhibited by beta-lactam antibiotics. The transpeptidase enzyme that contain serine is probably acylated by beta-lactam antibiotics with cleavage of CON bond of the beta-lactam ring. This renders the enzyme inoperative and inhibits peptidoglycan synthesis. So actually this peptidoglycan, it's also known as murin. So murin. So peptidoglycan is a polymer consisting of sugars and amino acids that form a mesh-like layer. So this mesh-like layer is present outside the plasma membrane of most bacteria, which forms the cell wall. So sugars like N-acetyl-muramic acid and N-acetyl-glucosamine are alternate and are connected through beta-1,4 glycosidic bond. Uh, you might have seen that the mechanism of action, a video, it's very clear in that. Please go through that if not seen.
Now we'll see the first one, that is penicillins. Penicillins refers to a group of beta-lactam antibiotics used in the treatment of bacterial infections caused by susceptible, usually gram-positive organisms. And they were discovered by Alexander Fleming in 1928 and produced by Penicillium species, mainly Penicillium notatum and Penicillium chrysogenum. The penicillin structure is composed of four fused four-membered beta-lactam ring with a thiazolidine nucleus. As you can see in the structure, it contains three chiral atoms, that is C3, C5 and C6, and 6-aminopenicillinic acid or 6-APA is the basic skeleton of all penicillins. So the R group decides the penicillin subtype, antibacterial activity, resistance to beta-lactamase and stability for stomach acids. The thiazolidine nucleus, which is represented by A. So as you can see, the structure of thiazolidine, it contains one sulfur and one nitrogen, and it is fused to a beta-lactam nucleus, which is designated as the letter B. So beta-lactam ring and thiazolidine nucleus. So these two fuse together to form the core structure of penicillins. And depending upon the R group, we have different types of penicillins.
Moving on to the classification.
First one is natural or biosynthetic penicillins. Examples: benzyl penicillin, 2-pentenyl penicillin, 3-pentenyl penicillin, N-pentenyl penicillin, N-heptyl penicillin, para-hydroxybenzyl penicillin, phenoxymethyl penicillin, and zero allergy penicillin. Second one, semi-synthetic penicillins. Examples: ampicillin, amoxicillin, methicillin, nafacillin, oxacillin, cloxacillin, dicloxacillin, flucloxacillin, carbanicillin, ticarcillin, piperacillin, meslocillin, cyclacillin. Penicillin combinations. Examples are benzathine penicillin and procaine penicillin. Second type, based on spectrum of activity. And that first one, those which are effective against gram-positive bacteria. Examples: benzyl penicillin, ampicillin, oxacillin. Second one, effective against gram-negative bacteria. Example: temozillin. Third one, broad-spectrum penicillins. Examples: ampicillin, amoxicillin, carbanicillin. Fourth one, penicillinase-resistant penicillins. Example: medicillin, nafacillin, oxacillin, cloxacillin, dicloxacillin, flucloxacillin. Fifth one, acid-resistant penicillins. Example: benzathine penicillin, phenoxymethyl penicillin, phenoxyethyl penicillin, phenoxypropyl penicillin. Moving on to the chemistry of penicillins. The structure of penicillin is composed of fused 4-membered beta-lactam ring with thiazolidine nucleus. So what is a thiazolidine nucleus? It is a 5-membered ring with 1 sulfur atom and 1 nitrogen atom. So thiazolidine nucleus. It contains 3 chiral carbon atoms at C3, C5 and C6. 6-aminopenicillinic acid or 6-APA is a basic skeleton of all penicillins. So we have seen already the structure of penicillin is composed of fused 4-membered beta-lactam ring with thiazolidine nucleus. So it contains 3 chiral carbon atoms C3, C5 and C6. And 6-aminopenicillinic acid is the basic skeletal structure of all penicillins. Penicillins differ from each other in antibacterial activity due to variation in structure of acid moiety of the amide side chain at C6. So you can see the amide side chain at C6. What is the amide side chain? C double bond O NH. So this is attached to the 6th carbon of the beta-lactam nucleus. So acylation of 6-aminopenicillinic acid with appropriate carboxylic acids result in new penicillin. Then structure of penicillins. We have already studied the basic skeletal structure of penicillins. So we have the acyl amino side chain RCONH. So we will see one by one. First one is penicillin G. Its chemical name is benzyl penicillin and the R group is benzyl group CH5CH2. Then second one, penicillin V. Chemical name is phenoxymethyl penicillin and the R group is phenoxymethyl group. Phenoxymethyl group. What is phenoxymethyl group? C6H5OCH2. So C6H5OCH2 is attached to the acyl amino side chain. It is attached to CONH, which is attached to the lactam, beta-lactam nucleus. Then third one is penicillin X. What is the chemical name? Para-hydroxybenzyl penicillin. So the hydroxyl group is present at the para position of benzyl penicillin. So para-hydroxybenzyl penicillin is penicillin X. Fourth one is penicillin F. Chemical name is 2-pentenyl penicillin. So 2-pentenyl penicillin. So 5 carbon atoms will be there. So CH3, CH2, CH double bond, CH, CH2. And in 2-pentenyl penicillin, the unsaturation is present at the second carbon. Okay, so 2-pentenyl penicillin is the R group present in penicillin F. Next we have penicillin K. Chemical name is N-heptyl penicillin. N-heptyl penicillin. So the R group will be CH3, CH2. Is it 6 times or 7 times? So total number of carbon atoms in heptyl penicillin will be 7. So in a textbook it is given wrongly. So instead of 7 it should be 6. So CH3, CH2 6 times is the R group present in N-heptyl penicillin. Then 3-pentenyl penicillin. So at the third position one unsaturation is present. So 3-pentenyl penicillin is CH3, CH double bond, CH, CH2, CH2. So this is the R group present in 3-pentenyl penicillin. Next one is N-pentyl penicillin. So CH3, CH2 4 times. Next one is phenethicillin. The chemical name is phenoxyethyl penicillin. Phenoxyethyl penicillin. Okay, so in phenoxymethyl penicillin we have CH5OCH2. So the hydrogen, one hydrogen will be substituted by a CH3 group in case of phenoxyethyl penicillin. The next one is propicillin. What is propicillin? The chemical name is phenoxypropyl penicillin. So how many carbons will be there? So total 3 carbons will be there. So phenoxypropyl penicillin, the R group will be CH5OCH, C2H5. Next one is methicillin. Chemical name is 2,6-dimethoxyphenyl penicillin. As you can see in the structure, 2 methoxy groups are present at second position and sixth position. So the R group in case of methicillin is 2,6-dimethoxyphenyl penicillin. Then naphacillin. Naphacillin contains 2-ethoxy 1-naphthal group. So 2-ethoxy 1-naphthal group. So a naphthalene ring, and at the second position of this naphthalene ring, an ethoxy group is present, OC2H5 group is present, and the first position of naphthal group is attached to the acyl amino side chain. Next we have oxacillin. The chemical name is 5-methyl 3-phenyl 4-isoxazolyl penicillin. So isoxazole nucleus is present in oxacillin. What is isoxazole? It's a 5-membered ring with 1 oxygen and 1 nitrogen. Okay, so the preference goes to oxygen. So numbering starts from oxygen of the isoxazole nucleus. So first numbering is given to oxygen, second to nitrogen, and the third to the carbon which is attached to the phenyl group, and the fourth carbon is attached to the acyl amino side chain, and the fifth is attached with a methyl group. So in oxacillin, the R group will be 5-methyl 3-phenyl 4-isoxazole nucleus. Next one is cloxacillin. So it's a side chain. The R group will be 5-methyl 3,2-chlorophenyl 4-isoxazole nucleus. 5-methyl 3,2-chlorophenyl 4-isoxazole nucleus. So the second position of the phenyl group is substituted by a chlorine atom. Okay, so fifth position of the isoxazole nucleus is attached with a methyl group. So 5-methyl. Then the third position of the isoxazole nucleus is attached to 2-chlorophenyl group, and the fourth position is attached to acyl amino side chain. Next we have dicloxacillin. So as the name indicates, it has 2 chlorine atoms. See the chemical name, it is 5-methyl 3,2,6-dichlorophenyl 4-isoxazolyl penicillin. Next one is ampicillin. What is ampicillin? It is D-alpha amino benzyl penicillin. D-alpha amino benzyl penicillin. Next one is amoxicillin. D-alpha amino para-hydroxy benzyl penicillin. A hydroxy group is present at the para position in case of amoxicillin. Next one is cyclacillin. It is 1-amino cyclohexyl penicillin. Next is carbenicillin. Carbenicillin is nothing but alpha carboxy benzyl penicillin. Alpha carboxy benzyl penicillin. Next is ticarcillin. It is alpha carboxy 3-thioenyl penicillin. So a thiophene nucleus is present. The R group contains a thiophene nucleus. So at the third position, it is being attached with CH-COOH. So alpha carboxy 3-thioenyl penicillin. Then next one is piperacillin. So piperacillin, a piperazine nucleus. What is piperazine? It's a 6-membered ring with 2 nitrogen at first and fourth position. Okay, so alpha 4-ethyl 2,3-dioxo 1-piperacenyl carbonyl amino benzyl penicillin. Okay, so next we have penicillin combinations. Under that we have benzathine penicillin and procaine penicillins. So, but benzathine penicillin is a combination of 2 molecules of benzyl penicillin with 1 molecule of benzathine. Benzathine is nothing but N-dibenzyl ethylene diamine. It is a white crystalline powder, odorless, freely soluble in formamide and DMF. Slightly soluble in ethanol, very slightly soluble in chloroform and water, practically insoluble in either, and it should be stored in a tightly closed container in a cool and dry place. It is given orally, intramuscularly or intravenously in a divided dose, and a single injection is found to be effective for a week. And now we have procaine penicillin. It is a combination of equimolar mixture of 1 molecule of benzyl penicillin with 1 molecule of procaine. It is a white crystalline powder, slightly soluble in water, sparingly soluble in ethanol, and it should be stored in a tightly closed container in a cool and dry place. It is given as intramuscular injection. It is a popular respiratory product which reduces the injection frequency. And its uses include used for the treatment of syphilis, anthrax, mouth infections, pneumonia, diphtheria, cellulitis, etc. And benzathine penicillin can be used for treating diphtheria, syphilis, and rheumatic fever. And in procaine penicillin, 1 molecule of procaine is present. Actually, what's the use of this procaine? It serves as a local anesthetic. Okay, so in procaine penicillin, which is also known as fortified procaine penicillin, it is given in combination form. It is 1 molecule of benzyl penicillin and 1 molecule of procaine. So this combination is aimed at reducing the pain and discomfort associated with a large intramuscular injection of penicillin.